So hello everyone. In our previous lecture we talked about mRNA degradation, and we discussed about de-adenylation dependent mRNA decay, where it was necessary in either case, whether you want to proceed in 3' to 5' direction or in 5' to 3' direction, it was mandatory that you first remove the poly A tail, and then you can degrade the mRNA molecule. So in both the cases, whether it is 5' to 3' degradation of the mRNA or 3' to 5', you first need to remove the poly A tail, and that's why it was called as de-adenylation dependent mRNA decay, because you could not destroy the mRNA molecule without destroying the tail first. Today we are going to talk about de-adenylation independent mRNA decay. So as the name indicates, that this is not dependent upon the removal of poly A tail. You do not need to remove the poly A tail in order to destroy the mRNA molecule. You can do this without removing without destroying the poly A tail. So the tail remains intact and you can destroy the mRNA molecule. So here we have example. There are not many examples for this type of mRNA decay. Here we have an example of an mRNA molecule. This is an mRNA for the ribosomal protein subunit 28B. So that's the protein part of the ribosome of the smaller subunit. So this is an mRNA molecule which codes this protein for the ribosomes. And this is a this works in a self-regulatory mechanism, that RPS28B has a binding site in its 3' UTR, where this RPS28B, this product of this mRNA molecule itself, can bind to its binding site in the 3' UTR. And this can recruit another molecule, what we call EDC3. So EDC stands for enhancer of decapping. Enhancer of decapping is a protein which can activate other factors which might be involved in degradation of the mRNA molecule. And in this case, EDC3 is going to recruit DCP1, DCP2 complex. And we have previously discussed this, that DCP1, DCP2 complex, this together constitutes, or both of them putting together work as a decapping enzyme, where basically DCP2 contains a mut T domain which can remove the 5' cap of the mRNA molecule. So after recruitment by the EDC3, DCP1, DCP2 complex, or this decapping complex, it removes the cap from the 5' end of the mRNA molecule, and this is followed by the action of XRN1, exoribonuclease 1, and it starts destroying the mRNA molecule in a 5' to 3' direction. We also have another example, an identical example from Saccharomyces cerevisiae for EDC1 mRNA. So that's also enhancer of decapping enzyme. So that would be the enzyme itself, means the mRNA of this enzyme. It also works in the same fashion. So both RPS28B and EDC1 mRNAs, both of these two mRNA molecules, they have the ability to get directly decapped without de-adenylation. They can bypass the de-adenylation step, and they can do so because they have a self-regulatory mechanism where they bind to their own 3' UTR, and they recruit an enhancer of decapping protein, ADC1 or ADC3, whatever, and these enhancer of decapping proteins, they have the ability to recruit other factors which can destroy the mRNA molecule at the 5' end, and this is how they achieve degradation of their own mRNA molecule. And basically there's in this 3' UTR, they have a stem loop structure. So if we can go to another slide, let's see if, yeah. So a stem loop structure is basically a secondary structure of the mRNA molecule, where the mRNA molecule can bind with its own nucleotides and can create a loop-like structure. That's what we call a stem loop structure. This is a secondary structure of the mRNA molecule, and these mRNA molecules for RPS28B or EDC1, they basically have this type of structure in their 3' UTR, and the proteins can bind to this stem loop structure. In the same fashion, you can also come across another type of structure in the mRNA molecules or in the RNA molecules. This is an nRNA pseudoknot structure, where you see rather than a stem loop structure. So this you can see that the RNA molecule binds at multiple sites with its own nucleotides and creates these double-stranded structures. And if this is much more complicated structure, it's not just a single binding between the two strands. So here we say that this is more like a pseudoknot structure, and this is the RNA component of the human telomerase. And these type of structures can have different types of impacts on the life of the mRNA molecule, as well as on the translational activity of the mRNA molecule. So they can either other proteins can recognize this structure and bind to these mRNA molecules to carry out certain functions, or they can have binding sites for other proteins which are regulatory, as far as which regulate their degradation process that we're looking at right now. So here we have these examples here. So for RPS28B mRNA molecule or for EDC1 molecule, so both of these mRNAs have these stem loop structures in their 3' UTR, and these proteins can bind to this stem loop structure. In case of this mRNA molecule, which is for RPS28B, the protein product of this mRNA molecule can bind to its own stem loop structure and then recruit EDC31, which is followed by decapping and degradation at the 5' end. Another type of mRNA decay we have previously discussed is that, so we have talked about the degradation at both the ends, at the 3' end, at the 5' end. We have talked about deadenylation dependent mRNA decay and deadenylation independent mRNA decay. I told you that you can also cleave the mRNA molecule in the middle by the activity of endonucleases. So this is the most efficient mechanism of killing an mRNA molecule, though not a very common mechanism for destroying the mRNA molecules. It's rather more efficient. And there are certain examples for the endonucleases like IRE1, PMR1, or RNase MRP. So these are basically the endonucleases which are involved in degradation of this mRNA molecule. Owing to their specificity, the specificity of these molecules, they are very well regulated. Otherwise, they have the ability to cleave the mRNA molecules, because the mRNA molecules are not protected in the middle. They are not very well protected, like they are protected at their 5' end by the cap and at their 3' end by the poly A tail. So both the 5' end as well as the 3' end are both of these two ends are protected by the are protected from the attacks of exonucleases. However, there's not much protection in the middle. So it would be easier for the mRNA molecule to cleave in the middle if these RNA molecules are frequently and easily available and functional inside the cytoplasm or even inside the nucleus. But they're very tightly regulated and they're very specific. I mean, if you talk about IRE1 and PMR1, so they're very tightly regulated and they're not very frequently available for their activity, and the same way RNase MRP, for example, this RNase molecule endoribonuclease is basically restricted to the nucleus as well as to the mitochondrion, so it is not going to do anything inside the cytoplasm. So that's their that's the restriction or regulation, that they're not functional in another location, and they're not frequently found inside the cytoplasm. Otherwise they would kill lots of mRNA molecules. So they need to be tightly regulated. So what happens after the mRNA molecule is cleaved in the middle? It can go either way, because it's unprotected in the middle now. So if you cut it here, so that should be the 5' end, and this should be the 3' end of the mRNA molecule. So this is cleaved into two parts. One part where which contains the cap at the 5' end, and the 3' end is naked. So we have previously discussed that the 3' end is attacked by the exosome, which is a complex of 6 different proteins with the RNA degradation activity. They have the capability to destroy the mRNA molecule, so they would start destroying it at the 3' end. So the decay would take place in 3' to 5' direction by the exosomes. While on the other hand, when the cap is removed, we have previously discussed that when DCP1, DCP2 complex removes the cap, this is followed by the recruitment of XRN1 to the 5' end of the mRNA molecule. So XRN1, the exoribonuclease 1, it starts destroying the mRNA molecule at the 5' end. It proceeds in 5' to 3' direction, destroying the mRNA molecule. So this is the most efficient mechanism of destroying an mRNA molecule, and several of these endonucleases, they're involved in cleaving the mRNA molecules in the middle, which is followed by both of these two processes, the 3' to 5' decay and 5' to 3' decay. So I think that is sufficient for today. So if you have any questions, you can post them on Google Classroom, and I would try to answer your questions. Okay, thank you very much for your attention.